Sam Pogosov, and I am privileged to be a guest host of Cognitum today. For those of you tuning in for the first time, Cognitum is a show in search of truth through science and technology. With us today are the exciting founders of new field of Immunodietica, systems immunologist Iosif Gerstein of Ajax Biomedical, and Dr. Leonardo Ferreira, immunologist at UC San Francisco. Gentlemen, welcome.
Thank you.
Tell me, what is Immunodietica?
So Immunodietica is a systematic study of the interactions between diet and autoimmune disease.
And autoimmune disease is?
Well, so the immune system is usually seen as an army that protects us against viruses, bacteria, and cancer. But sometimes the immune system also goes rogue and attacks your own tissues. It's like the body at war with itself. And so in autoimmune disease, your immune system will recognize and kill your own tissues. For example, in type 1 diabetes, it will recognize and destroy your pancreatic beta cells, which make insulin. And so you depend on injecting insulin for the rest of your life.
It makes sense.
Fundamentally, it's a confusion of self and non-self. A well-regulated immune system can tell the difference between self and not to attack it, and non-self, in which case it's a foreign invader or potential pathogen, and attack it. And when there is a confusion, when it recognizes an epitope as not self, that's self, then there's the generation of autoimmune disorder.
In simple terms, what is an epitope?
An epitope is a sequence of amino acids, 8 to 20 amino acids long, which is somewhat like an ID card that tells the immune system whether this is a cell that is self or not self.
That makes a lot of sense. So basically, a cell holds an ID card, and the patrol system either treats it as a friend or a foe.
Correct. And so this ID card is derived from proteins inside the cell. So if you have just a normal cell, it will just present its own proteins in this ID card on the surface. But if you have a viral infection, for example, now the virus will be inside the cell making its own proteins, but then these proteins will be presented on the surface. So the immune system will come and will say, oh, this cell has viral peptides, viral epitopes on its surface. We must kill it. In autoimmune disease, a beta cell from the pancreas will have an insulin-derived peptide on the surface, and the immune system will erroneously recognize and kill, hence the term autoimmune disease.
That sounds like a pretty important challenge to solve. How are you fellows going about it?
Well, we came up with a theory that certain foods you eat can create confusion in the immune system. So if you eat tissues that are similar to your own, then possibly this can lead to the development or increase in severity of symptoms of autoimmune disorders. And so what we did is we looked at roughly 18,000 autoimmune-associated epitopes in the human immune system, and we cross-referenced them with about a dozen commonly consumed foods, now roughly 24, to see where the overlap is.
What do you think about the possible mechanisms between diet and autoimmunity?
So this is actually a very interesting question, because one would assume that when you eat something which has just protein, you just destroy that protein completely when you absorb the amino acids. But that's not completely true. For example, some people are allergic to certain foods. So some people who have peanut allergies eat peanuts, they mount an immune response and actually die from eating peanuts. So clearly the immune system can see epitopes derived from food that you consume. And so what happens actually is that your gut usually does not let bigger proteins go through. But sometimes it does. It's called a leaky gut. And that's caused by inflammation. Actually, it's been shown that if you cause inflammation even in the skin, you can activate immune cells that will communicate to your gut. It'll increase the gut permeability. And so now you're exposed to proteins, to epitopes from food in an inflammatory context. And the immune system is all about context. If you have one protein presented in a peaceful context, so there's no danger signal, we call it, T cells, which are the main killers of the immune system, they don't do anything. Actually, they become anergic, which is what we call T cells are just sleeping, resting peacefully. But if you have the same exact protein epitope in the context of danger and inflammation, which could be some pro-inflammatory cytokines, could be some microbes, could be excessive sugar or salt in your diet, then it's a different story. Then your immune system gets activated. And so just like when you prick your finger and there's a cascade of pro-inflammatory molecules that churn up and then call all immune cells to that site, likewise in your gut, you have immune cells that get recruited to the inflammatory sites and they have this protein. And so now, what if this protein, this epitope, is an exact match to one of your epitopes of your tissues? Once the epitope goes away, your T cells have been activated already. So even though that a local immune response will cease, you now create memory T cells. These T cells remember that they saw this epitope, and now they'll travel to your body, and next time they find that same exact epitope that they are specific to, they will attack again.
With or without the context?
With the context.
With the context.
And so, for example, another interesting piece of data is that many people have autoimmune disease. They only develop it after certain viral infections. And so what is thought is that, once again, these viruses have epitopes that are exact matches to an autoimmune disease-associated epitope. And so the person is just fine. It does have autoreactive T cells. So these T cells are the bad guys. They escaped education, and they somehow still see your self-tissues. But if there's no danger, they don't do anything. They don't really cause any trouble. But if you have a viral infection, you now have the epitope plus the danger signal. And so, yes, you can clear the viral infection, but your T cells, your memory T cells, they have now been activated, they remember it. And so next time they go around, if this virus has an epitope that matches a pancreatic cell epitope, and your pancreas gets inflamed, you now, unfortunately, have T cells that can go and attack your pancreas.
Attack
and so that's
your own pancreas.
And the reaction keeps going, and then you get type 1 diabetes.
It's fascinating.
So this sort of transient leaky gut syndrome can be one potential mechanism of how diet does interact with autoimmune disorder.
That makes a lot of sense, even on a very, very simple level for me.
And so this is why we think that, again, really mapping all the possible epitopes that look just like our epitopes. And again, we applied a very stringent filter. We didn't just look at all human epitopes. We looked at human epitopes that have been implicated in human autoimmune disease. So we know these are dangerous epitopes. You don't want these epitopes in your body because they've been implicated in autoimmune disorders. And so, once again, we hope that little by little we can build a very comprehensive platform that can really help people with autoimmune disease and even the population in general.
Because if you eat one of these proteins and it gets broken down into peptides and then it goes into your bloodstream through the leaky gut, these peptides will have epitopes. And if those epitopes coincide with the autoimmune,
you get the patrol coming in.
The patrol comes in, and that's basically the mechanism that can cause this interaction. At least that's one potential way.
Yeah. It makes a lot of sense. What were some of the findings?
Well, so interestingly, we found that if you look at these 14 different commonly consumed species, one of them really stands out because it's got almost 1,000 epitopes found in human autoimmune disease that you don't see in any other species.
What species is that?
And that is the pig. So the pig has almost 1,000 epitopes. They have been previously implicated in almost 70 different autoimmune disorders.
And this is particularly interesting because when we were developing the theory, our initial hypothesis was focused on the pig. And it was just under 2 years ago when me and Leo were discussing this topic in Las Vegas, and we were talking about the problems associated with cannibalism and the prevalence of autoimmune disorders in those societies. And I thought, well, one, we have cannibalism having higher prevalence of autoimmune disorders, and two, so many cultures have prohibitions on eating pork, including the rules of kashrut, my own faith. And therefore, the initial hypothesis was there might be something to it, that because this rule has lasted in so many cultures and for so long, it's lindy. It has some truth value or it has some fitness value to it. And lo and behold, when we actually got through the data, we came to this completely unexpected conclusion, which we had initially hypothesized based on the theory.
You mentioned cannibalism. Why?
Well, the reason I mention cannibalism is because pigs are one of the only animals that we eat often and that are suitable for transplantation. So eating organs of pigs seems to be one of the closest things just I had thought of on a visceral level back when we were developing the theory to eating human. And there was some anecdotal reports, I don't remember if it was online, I don't know where I read this, but that pig meat tastes like human meat. Now, I don't know the source, I don't know how one could figure out that source, but these factors led me to consider some similarity. And the similarity of the immune profile of the pig with the immune profile of the human was more aligned than could have been predicted genetically. So there's something there.
In addition, it has been shown that people that work in pork facilities where their job is to blow out pig brains for processing to sell, some of these workers develop autoimmune disease in their nervous system. And so they later did research and found out that the pig epitopes are so close to ours that these people inhaled aerosolized pig brain antigens and they developed an autoimmune disease in their nervous system. And so they found that some of the epitopes in the pig were close enough to ours that these people developed in weeks what usually takes many decades to develop disease, an autoimmune disease of the central nervous system.
Has something similar happened in beef or poultry processing facilities, or just the pig?
Not that has been reported. So no data is not the same as negative data, but so far the pig is the one case where we saw this dramatic. It was, I believe, was almost 100% of the workers eventually developed this autoimmune disease by being exposed to pig brain antigens in the air.
It certainly sounds like there's something there. What is the future of Immunodietica?
Well, so one thing that we really want to do is to incorporate data from individuals. So for example, different HLA alleles, and HLA are these billboard proteins on the surface of cells that present the ID cards, the epitopes. So everyone has a different combination. We get an allele from mom, an allele from dad, and there's 5,000 alleles for HLA-A, 6,000 for HLA-B, 5,000 for HLA-C, and so on and so forth. There's like 8 total. So if you do the math, there's like trillions of possibilities. And so all of them will play in a slightly different way with the epitope, right? Because it's a molecular interaction where the epitope sits on the growth of this MHC molecule. And so that's one thing that we really want to incorporate in our platform. Secondly, we also want to study the microbiome, because it's been published that different microbes interact with the immune system differently. So different compositions of your gut microbiome will preferentially feed and foster different types of immune diseases. Actually, even salt intake has an impact on your immune system composition. If you eat too much salt, you have an upregulation of Th17 cells, which are a pro-inflammatory T lymphocyte subset. And so all these things will, with time, be incorporated as additional layers to our Immunodietica platform.
Another very, very important aspect of our work is the potential to isolate targets and understand the mechanisms of autoimmune disorder better and potentially find new avenues for therapeutics. Because if you can isolate unique epitopes that in certain animals cause flare-ups of symptoms, which we could find out if we got feedback from our users, then there could be a targeting of trying to understand the mechanism, and if you can target the mechanism, then you can start engineering therapeutics around that.
And most autoimmune disorders do not have a cure. And so really it's an open field to attack this disease from several angles. And so once again, because the environment seems to play such a proportionate role in autoimmune disease incidence and severity. Like for example, in rheumatoid arthritis, people say that certain foods will cause flare-ups of the disease and others don't. And also between different people, the answers will be slightly different.
Right.
And so we hope that with our platform, we can really help these patients and more in really understanding and making wise choices in what to eat and what not to eat.
That sounds fascinating. What are some of the steps forward for additional, perhaps, research or additional findings?
Well, so next, as Iosif already said, we're going to look at more species that are commonly consumed. Secondly, we're building this platform to systematically dissect these interactions between diet and autoimmune disease. And I should actually say that autoimmune disease, it seems to be 70% or more caused by environmental factors. So the study of diet and autoimmune disease actually is very important because it's one major environmental factor that causes this disease and can also modulate its severity. But so that's one step, adding more species. Secondly, we also want to take data from the individuals. So for example, these billboard proteins that present the ID cards, well actually all of us are different in the sequence of these genes. They call HLA, human leukocyte antigen. And so they are the ones that really make us different at the immunogenetic level. Like if I transplant an organ from me into you, it will be rejected because it's different, it's non-self. And so different people will respond differently to these same epitopes derived from the same food. And so that is something that we want to build onto our platform to analyze this data.
That sounds great.
And it presents a host of computational issues in order to generate these databases. Because just with our 25 organisms, we already have more than half a million data points in our database, along with the combinatorics associated with immunogenetics. It starts being very, very important to find the right epitopes to focus on, the right genes to focus on, in order to create actionable insights for autoimmune patients.
And it sounds like you might need perhaps a supercomputer or two to really crunch to the data.
Well, fortunately, with the cloud revolution, we have access to virtually infinite infrastructure. It's just a question of scaling the algorithms, setting up the systems, and executing.
I think with this, we can really bring about this personalized diet advice for autoimmune patients and the population in general. Because as of now, many books have been written about diet tricks and tips for autoimmune disorders, but we think our approach to autoimmune disease is based on immunology. And so we are using immunology to understand the impact of diet in autoimmune disease. And so we believe that is a dramatically different approach to this, and hopefully one that allows it to gain deeper understanding of what's behind these mechanisms of how diet interacts with autoimmune disorders.
I understand it's still early in the process. You guys have just really released some of these findings. But what are you hearing from the scientific community in terms of support or direction, guidance?
Well, quite recently we were published in the Journal of Translational Autoimmunity. That's our first paper on this project, and the response has been largely positive from correspondents.
We also, before even the official paper, we had a preprint out, and we were almost flooded with emails from people asking more about different questions about what we're doing, what we want to do next. And so we both published our paper recently. We also spoke at a conference on immunology. And so really, I think we're moving pretty fast and we try to dissect more and more what could be these interactions between diet and different autoimmune diseases.
Yeah. I heard the term the Gershteyn-Ferreira index, the GF index. Tell me just a bit of the mechanics of how it works and how it represents the metric.
So the Gershteyn-Ferreira index is a simple to use measure for autoimmune patients, of which there are about 50 million sufferers in the US alone, which could give them, for their disease, our ranking of the potential for immunogenicity of different foods. What I mean by that is we've simply taken the overlapping epitopes of the different foods without any personalization, and just given an overall global picture of how those foods rank in terms of overlap. And then we normalized that. And we did that on a disease level, and we did that on a general autoimmune level.
So as an example, for example, I know folks know the glycemic index is 0 to 100. And it's somewhat simple now these days to say this food has a GI of 55, so I know how much is going to trigger my blood glucose level in sugar. Sample of the GF index would be a 0 to 100, or would it be 0 to 10?
0 to 1. That's why it's normalized. So we tried to make things as simple as possible. So we created 3 levels of the index. We created a global index, we created a disease-specific index, and we created a unique index. What that means is on a global index, the theoretical maximum would be a 2. But because human is not a commonly consumed organism by humans, it's normalized to the largest in-sample organism. So that would be a 1. On the disease-specific, it's also normalized to the highest to be a 1. And on the unique, the 1 is actually the pig.
Okay, so pig would be a 1, and let's say celery would be 0.
Celery is out of sample, but likely to be negligible.
But some vegetables were close to 0. So a non-zero value, but still very close to 0.
So basically using the index of zeros and ones, you can assign certain value to particular foods and organisms and use that to benefit people's diets.
Exactly. And what we can do right now is that people can go on our website, Immunodietica.com, and they can look at their disease and find out which foods are more dangerous and which foods are less dangerous for their disease. In the future, what we hope to do is to be able to integrate a lot of personalized data, genetic data, biomic data, and have a platform that creates individualized dietary recommendations based on this individual knowing his information.
If our audiences are consuming certain foods that aren't currently in your field of, on your radars, can they submit perhaps over time certain things they eat and help you build your database and be engaged with your process?
Absolutely.
Yes.
What we're hoping to do is to grow this database as resources and time permits. We've already almost doubled our count since the publication of our paper, and we expect to continue to do so.
They say whatever happens in Vegas stays in Vegas. You guys mentioned that the idea, the theory, appeared to you, you guys came up with it in Las Vegas. What happened after? Tell us a bit about the process.
Well, we came up with the theory in Vegas after discussing it for pretty much the whole time we were there. And the enormity of the task didn't really hit us until we started creating these computations. There are over 70 autoimmune diseases that are in the database. We started our first papers on 14 organisms. There are 18,000 human epitopes associated with autoimmune disease. And so every combination of every one of those had to be investigated and be part of the database. So automating that system for the database creation and then analysis was a pretty large task.
It's a massive task.
And every step had its own problems associated with it. And it seemed that the combinatorical problems associated with almost every step had to be solved in a very interesting way, otherwise it's a deal breaker. And we got quite lucky in making sure that our methods of data collection were lean.
In other words, instead of trying to muscle through and boil the ocean on this, because it sounds like it's a lot of combinations and permutations, you had to devise a more elegant approach on how to tease out these insights.
These and more, we didn't stop there. We also asked, where are these epitopes expressed in the animals and plants? Are they expressed in the liver, in the kidney, in the brain, everywhere? And so we actually found that from the many pig epitopes uniquely found there that are being implicated in human autoimmune disease, many of them are actually expressed everywhere. So when you eat pork, you are exposed to these epitopes. They're not just in the brain or just in a specific organ, they are everywhere. And so that's obviously especially important. And so that took, once again, a big effort computationally, but also manual annotation, which you really go and make sure that the data really makes sense. And so many of these proteins, I know them personally, if you will, and so I know where they are expressed. And so there's a lot of computational effort, also of manual annotation. So we mapped the epitopes in species and autoimmune disease. Then we investigated where they're expressed, which tissue. And now what we're actually doing, and so this part is in our paper that was published, actually, by the way, this paper is open access. Anyone can go online and read it. Just Google Immunodietica. We're the first and only results on Google. And on top of that, we're now actually moving on to mapping these epitopes, not just where they're expressed in the body, but also to which HLA molecules do they bind to. Because once again, people have thousands of different HLA alleles. Every person only has like 6 of them, right? And so if we can really map these epitopes to specific HLAs, we can now really take this global platform to a personalized tool that we can adjust to every person individually. And so that's the current computational effort that is even bigger than what Iosif just described. That's several orders of magnitude bigger effort. Then we might really need a supercomputer to process, because this will really be millions of data points just to test the 500 epitopes that are combined between different species uniquely. Just for those 500. Now imagine 18,000.
Right.
Now imagine different people.
Right. Now, I'm sure you guys thought of this, but on the simple level, studying groups, there are certain tribes, folks who eat pork, those who don't eat pork. Taking these groups and teasing out some top level insights. Have you found anything that's pervasive?
So epidemiological data has not been part of our first paper. We're actively looking for an epidemiologist to coordinate with on a future paper. However, when you're comparing different populations, you have so many variables, and the HLA is just one of them. So until we have a more comprehensive map of how genes interact with autoimmune disorder, that layer of society or subpopulation level dietary differences is likely to get washed out in the data.
Makes sense.
So until we have a control for the deltas in the immune system through the genetic component, the deltas on a social level, not on an individual level, on general consumption patterns is going to be a very, very difficult path to pursue, but we are very interested in pursuing it.
That makes a lot of sense. How can the scientific community who's going to be exposed to your papers and your work in the conference that you were invited to, how can they help you accelerate this exploration?
Well, collaboration, definitely. We're looking for collaborators to co-author new studies on investigate any sort of mechanism that can be uncovered, and we try to make all of our data that we published in the paper available to any researcher who's interested.
Yeah, science moves faster as a team. Right. And so an epidemiologist, for example, which neither of us is, would be a great addition, for example, and you can go so on and so forth. And because I think this effort will really just continue to grow and to bear fruits. You really need to make a team that can really explore every facet of it to really continue making progress.
And I also, being from Boston, and Dr. Ferreira, you're from being in California right now, you guys do really have the coast covered. So yeah. I think folks can really, you're accessible. That sounds like you're accessible for other scientists and other like-minded individuals who can jump on and help the cause. Yeah.
I think we're trying to be proactive in reaching out to individuals who we think might be interested in the work, and we're hoping that there will be more collaborators as our work grows. Immunology is in general a growing field, and more related to almost every aspect of human well-being, all the way from depression with the inflamed mind hypothesis to cancer with the alternative hypothesis of cell respiration. So fundamentally, diet impacts the immune system in a grand way, and so it's going to impact general health and well-being probably in ways we can't imagine right now.
As a wise man once said, let food be thy medicine, and thy medicine be your food. And you gentlemen have given us a lot of food for thought today. So thank you very much for the information. I personally learned quite a lot.